Thank you very much. I would like to thank the organizer for the kind invitation, and it's my first time at this conference, but I greatly enjoyed the week, and it's been extremely interesting. So my name is Mara Fornaro, and I work in the disease of aging and regenerative medicine at Novartis. So I'm Novartis employee, and I own Novartis stocks. So I think we have heard every day, I think, this week about the Great Wave, meaning the significant increase in the aging population. And we all know that by 2050, 2.1 billion people will be age 60 and above. And already in 2017, 51% of the global disease burden was accounted by age-related diseases. Many of these age-related diseases are on strategy for Novartis. So I guess it doesn't come as a surprise that our department, the mission of our department is to elucidate and target the molecular mechanism underlying disease of aging and to develop regenerative pharmacological interventions for patients. So we are not new to this kind of work, and in the past 20 years, our department has worked to tackle musculoskeletal diseases of aging. We have brought a drug candidate into clinical development. We have heard about one of these candidates. Actually, David alluded to it yesterday, bimagrumab. But today I will not talk about bimagrumab, but rather I would like to give you a few highlights on different programs that we have. The first one is LNA043, which is a therapeutic protein that we are currently testing in patients with osteoarthritis. And then I would like to change gear and and give you a few highlights about the exploratory neuromuscular research that we have done and that has led to uncovering the therapeutic potential of targeting inflammatory microglia in aging. And finally, I would talk a bit about the SIGA-STIM pathway and its dysregulation in aging and peripheral nerve injury and its emerging relevance in diseases of aging. So starting with LNA. LNA is a therapeutic protein that was discovered using our regeneration engine, looking for factors that induce chondrogenesis. So LNA was shown that not only it induces chondrogenesis in vitro, but was also shown to regenerate cartilage in cartilage injury and repair model in the mini pig, as shown in the top left of this slide. But I think more importantly, LNA was shown to promote hyaline cartilage regeneration in patients with focal articular cartilage lesion after one intraarticular injection, and those are the data shown on the right side of the slide. Age-related changes during aging predispose to development of osteoarthritis. And there are currently no therapeutic options or treatments for osteoarthritis. So since we had shown that LNA was efficacious in cartilage regeneration, it was a logical step to test it in rat model of osteoarthritis. And as you can see at the bottom left of the slide, LNA treatment was also able to regenerate cartilage in OA model, in OA rat models. And in 2021, Novartis received a fast-track designation for LNA, osteoarthritis of the knee, and as I said, clinical trials are undergoing, so stay tuned and for the outcome of these trials. And now I would like to change gear and talk about the exploratory neuromuscular research that was carried on mainly by two former postdocs in our department. And as we have heard in the past few days, the neuromuscular system, of course, is also subjected to age-associated changes. And one change that is quite detrimental is the decline in motor units, which is a major factor, as we have also learned yesterday, in the development of sarcopenia and in the decline in mobility and fine motor control in aging. And I think the good news is that it has been shown that exercise has beneficial effects in the neuromuscular system, and therefore there are ways to prevent this decline. So Elisa Giorgetti, this is mainly the work of Elisa Giorgetti and colleague, was a postdoc in Mark Nash lab, and she wanted to study age-associated changing in the neuromuscular system. And she decided to do that by analyzing the decline in motor unit numbers in mice over time. And as you can see on the top left, in mice, there is quite a significant decline starting at 10 months of age and then up to 18 months. And importantly, I think, by performing transcriptomics in the spinal cord, she found that this altered muscle innervation coincided with spinal cord gene expression changes. And Elisa found that there were 17 genes that were upregulated in aged mice compared to adult mice. And many of these genes are expressed by the microglia, so the immune cells of the CNS. She also found that there was a significant enrichment in microglia signature in aged versus adult mice. And then by performing histological analysis and looking at the expression of IBA1, which is a microglia marker, she saw that the IBA1 expressing cells, where the number was upregulated in the spinal cord of aged animals, and that also there was an upregulation in the spinal cord in the expression of microglia activation markers. So she then wanted to explore and see if exercise was able to reverse these age-associated neuromuscular changes. And so she subjected mice to voluntary wheel running for a couple of months. And what she saw was indeed, she confirmed that the exercise can reverse the decline in motor unit number that is observed in the age mice, and also importantly, exercise was able to downregulate the upregulated microglia signature in the aged animals. So I think this data indicate that probably during aging, microglia engender a neurotoxic environment that is negative for neuronal health, and therefore she asked if we now deplete microglia, can we reverse the neuromuscular changes associated with aging? And to do that, she used an inhibitor of colony-stimulating factor receptor 1. This is a receptor that is extremely important for microglia function. And it's an inhibitor that was developed at Novartis originally for oncology by our oncology department. Is very specific, and as you can see, regardless of the administration regimen, the inhibitor was able to restore the motor units loss in the aged animals. In the meantime, and there were also other groups showing the beneficial effect of modulating microglia, and therefore this is now considered a therapeutic option in neuromuscular diseases. In addition, another colleague in the department, Mike Bidinowski, tested the inhibitor in a mouse model of ALS and showed its efficacy, demonstrating also the therapeutic potential of modulating microglia, inflammatory microglia in neurodegeneration. So as we all know, and as we heard in the past few days, an hallmark of aging is also impaired regeneration, and this affects several tissues. In addition, we have also chronic inflammation that might also impact regeneration. So Elisa set out to analyze the impact of age on regeneration following sciatic nerve injury. And then, as you can see on the left side of the slide, she showed that indeed there is a delay neuromuscular recovery with aging following sciatic nerve crash, as measured by analyzing compound muscle action potential and motor unit number, as well as by MRI. So another postdoc in my lab, Giulio Morozzi, was interested in understanding the molecular mechanisms associated with peripheral nerve injury during aging. And so to do that, he did a transcriptomic profiling of the sciatic nerve before and after sciatic nerve crash. And as you can see in the right side of the slide, what he observed was that there is a very fast upregulation of the inflammation and of genes that belong to the inflammatory pathways and the innate immunity response. And many of these genes that were upregulated belong to the CGAS STING pathway. However, as you can see, the upregulation of these genes declines with time in the young animal, but it remains sustained in the old animal, indicating either a lack or a delayed resolution of the inflammatory process in the aged animals. So I just wanted to say a few words about the CGAS STING pathway. As you probably know, CGAS STING senses cytosolic DNA. It recognizes foreign DNA, but also self-DNA, so nuclear or mitochondrial DNA that is released in the cytosol. And the sensor, the DNA sensor, is CGAS. And when CGAS binds to the DNA, basically then generates a second messenger, cGAMP, that activates STING, and STING triggers a type 1 interferon-mediated response. So Giulio wanted to see the role of STING in peripheral nerve regeneration in the aged mice. So to do that, he subjected wild type and sting knockout animals to sciatic nerve crushed, and then he measured the recovery of the compound muscle action potential post-injury. And as you can see, he confirmed that there is a delayed recovery of CMAP in the old mice compared to the young mice. But also what he noticed was that in the old animals, where sting was ablated, there was a faster recovery of nerve conduction. Now, he then wanted to see if there were changes associated in the microglia, in the spinal cord, in the animals that were, in the aged animals that were in the wild type and knockout animals. And as you can see on the bottom left in the slide, he found that there were many more activated microglia in the wild type as compared to knockout animal 4 weeks post sciatic nerve injury. 1 week, sorry, post sciatic nerve injury. So therefore, we hypothesized that cGAMP is probably released either by sensory neuron in the dorsal root ganglia and then transported to the spinal cord where it can activate microglia, or the other possibility is that the motor neurons in the spinal cord might actually release cGAMP and then activate microglia. And to show that microglia was indeed activated by cGAMP, he took advantage of BV2 cells, which are basically brain microglia, and he treated the cells with cGAMP over time, and you can see that cGAMP induced quite a significant increase in inflammatory cytokines, and this induction was ablated, prevented by sting knockout. In the meantime, in the past few years, other groups have confirmed that the cGAS sting pathway is upregulated in different organs, and we also have shown that, have seen that the type 1 interferon signature is not only upregulated in aged versus old mice, but is also upregulated in a mouse model of ALS and also in patients, in ALS patients. And now there is emerging literature showing the relevance of this pathway in different diseases of aging, and therefore the idea that targeting this pathway may provide therapeutic benefits in aging and age-related diseases is emerging. So I would like to conclude just by saying that, as I said, the mission of our department is to restore cell and tissue function in diseases of aging, and we would like to do that by targeting specific cellular processes, cell fate, and rebalancing cellular states. I alluded to the, for example, inflammatory microglia. And integral in this work, very important for our work, is integration of omics data sets especially from longitudinal studies performed in human subjects or even also in animal models. So, and with that, I would like to thank all the people that have contributed to the work that I've just showed you. And I would like to thank you for your attention, and just remember that we have an open position in our department, and if you're interested, please reach out to me or calling. And again, thank you for your attention.
Thank you so much, Mara. We have a question down there.
Hi. Have you looked into using blood flow restriction in any of the models to reverse sarcopenia?
Caloric restriction?
Blood flow restriction.
Ah, blood flow. No, sorry. No, we have not.
Okay, thanks.
Lan, which cell it's regenerating, and does it have regeneration capacity for other cells? Like,
No. So the regeneration was only tested in chondrocytes. We have not tested other cells, but this is a very good point. Yeah.
Thank you for the great talk. I'm just interested about the exercise that you talked about. Did you look at sort of the length of exercise, of the time of exercise? Is it correlated with, I guess, reduce of the disease, of the neuromuscular disease? Or you just did exercise versus no exercise?
We did exercise in aging, so in animals that were aging. We didn't relate that to any disease model, if that was the question.
We will take it. We'll take it after. Thank you so much. That was an amazing talk. Okay, maybe the co-organizer, Alex. Wait, Mara.
One question. By the way, for those of you who don't know, like CGAS STING pathway is like super hot right now in pharma, right? So everybody is developing it for cancer. Everybody is developing it for many other diseases. What do you think about Novartis' strategy and other strategies for dual-purpose therapeutics, for like CGAS STING inhibitors or agonists? And also, what do you think about ENPP1?
Let's start with the last one. Honestly, I don't think anything because I don't know much about it, so I cannot talk about that. Regarding the SIGA-STING pathway, I agree with you. It's a very hot topic, and a lot of companies are interested. And there are STING inhibitors that are being developed, as we know. And of course, this is something that it's of interest.
And you are using Novartis inhibitors for that, right, I guess? Or
no, we don't have Novartis inhibitors for that at the moment. But I mean, I can say probably because it was in the news, Novartis has acquired a company that has developed a STING inhibitor.
Of course.
All right.
Congratulations.
Thank you so much, Mara. That was really fantastic.